So primarily when a patient approaches us, and I'm a geneticist, that's my background, I'm a medical doctor, but specialized in genetics. The first thing we actually do is to try to understand the clinical situation of each individual patient. So for example, if a patient approaches us with a disease we call glioblastoma, which is a very malignant brain cancer, we hear about the clinical history, and then we ask when the surgery took place or the biopsy, because for our diagnostic step we need tumor tissue, and it's crucial to have the most recent tumor tissue. So for some patients a biopsy is a while ago, but especially when you design personalized treatment, you need to have a very recent tissue. Then we get a blood sample from each patient, because what we are doing in the end is a comparative sequencing. So we compare the normal genome of each patient with the tumor genome. And then most importantly, we need the informed consent of a patient and coverage of costs. And we can talk about this later. From a technical perspective, we isolate the DNA from the leukocytes, from the blood, and from the tumor. And also we isolate the RNA from the tumor. And then we use a very comprehensive sequencing approach, which we call whole exome and whole transcriptome. And in the end, what Darren was referring to is what are the unique mutations that we find in the tumor. Because every human carries mutations, we are different in about 3 to 4 million positions in our genome. But what we need to understand is what is unique to the tumor. We need to understand what is driving the tumor to grow, and these mutations, these are the ones that we are interested in. And we have two different groups of mutations. One group we classify as driver mutations, so they are driving the growth of the tumor, and the other class we call passenger mutations. So they are riding on the surface of tumor cells, and they are presented through HLA. And now what we try to find out is the number of what we call neoantigens. So the mutations that are unique to the tumor that are presented through HLA, because these mutations can theoretically be recognized by T cells. And these are the cells that we are interested in when we are talking about the immune system and about personalized immune therapy. So our approach, sequencing the tumor genome in comparison to blood, identifying the neoantigens, is what we do in a very first step, which usually takes about 4 weeks. And then the question of Darren was, how do we define the list of neoantigens that is unique for each individual patient, because this is the recipe that we can use in order to manufacture a personalized vaccine. And how do we prioritize these neoantigens? If you think about the number of a mutation load in a tumor, you have large differences. For example, if you take a malignant melanoma or a lung cancer from a smoker, you have hundreds and thousands of mutations. If you, for example, take a brain cancer from a child, you have a very small number of mutations. And then you have a very large spectrum. And the number that we use in order to define the tumor mutational burden is called TMB. This is the number of mutations per megabase, so per 1 million positions in the tumor genome. And depending on this number, we have a larger or a smaller set of, in the end, neoantigens. So the neoantigen is defined as being unique, is being defined as presented through HLA, and we need to know the likelihood that this mutation is on the surface of the tumor and can therefore be used for a personalized immune therapy strategy. And here lots of algorithms are used. They are either published and developed in-house, and then we have a large database of immune data. So once we do a personalized vaccine in a patient, we can, after certain time, usually it's about 3 months after the first vaccine, we measure the T cell response that is specific towards a neoantigen. And here we also have a large database. So using databases, using different algorithms, we can come up with a list of neoantigens that we prioritize for each individual patient. And here we also filed several patents because it's not only about the list of neoantigens, but it's also about the knowledge you need to have on the solubility of peptides, on the likelihood they all go in solution, they are stable in solution, they are then basically presented by antigen presenting cells, and the likelihood you induce an immune response. And this number, the likelihood of inducing an immune response for our approach, is nearly 90%.
Okay. Saskia, you've been doing this for a long time at Seagat. Can you talk a little bit about what the success rate is? Do we have any data on how well this has worked outside of glioblastoma?
Well, that's an important question, and it's difficult to answer because when a patient approaches us, it's always a unique situation. The majority of patients approaching us are late stage 4. They have been running out of standard options. They have been running out of guideline therapies, even off-label therapies. So they are, by definition, in a not so good situation. And then it's hard to tell you an exact number because there's a broad spectrum of different tumor entities. The majority of our patients have glioblastoma, and this is what we just published last August in Nature Communication. And there you can see that we provide evidence for the finding that if a patient develops an immune response, that they have longer overall survival compared to patients without immune response. But there's nothing is standardized. It's not a clinical trial. It's a real world observation, which makes it so difficult. And I'm not pretending that we are really having here a causal effect, but what we see is an observation, and this is now tested in the clinical trial that was mentioned in the introduction.
Okay. We've been reading a lot in the papers over the last year or so about the mRNA vaccine, which I think is similar, but how does your neoantigen peptide vaccine differ from that?
So if you think about the process of presentation, so if you go from DNA to RNA and translation into protein and then presentation through HLA, you might notice that the peptide vaccine approach is more direct because when you synthesize peptides, inject peptides in the skin where you find antigen presenting cells that basically take up peptides and present to the immune system, this is much more direct compared to bringing in the coding information through an mRNA vaccine that needs to be translated within a cell, that needs to be processed in the cell, presented in through the cell. This is much more complicated. So our process in general is more direct. And then the question is about immunogenicity, and there's no data of comparison. So ideally, you would have the same patient with the same tumor and the same mutation, and you immediately see this is not possible.
Right.
I think we have to rely on what is published by other groups, and then we can especially see how the long-term effect compares. From peptide vaccine, it's known that you can induce a long-term immune response, even for years, up to 10 years, an immune response could be measured. We have to see whether this is true for mRNA. We actually know from COVID that this doesn't look so great. So we will see.
I had another question or comment about that. I talked to CureVac, which I understand is like your neighbor. You work directly next to CureVac. Miriam and Dilla, Marcus Bergman, they were talking to me, and it sounds like they have a mRNA GBM vaccine based on antigens that are common to pretty much all patients. There's like they found there were like 9 epitopes that are pretty typical of glioblastoma, and so it would not be based upon sequencing anything. You would just have an off-the-shelf vaccine that works for all patients, because those suppose like survivin is an example, like BIRC5 occurs in quite a lot of cancers and quite a lot of glioblastoma. And so I didn't know whether you use these kinds of antigens that are not neoantigens, but just tend to be found on glioblastoma in general, whether that's effective or not. And of course, they are also doing mRNA as the modality rather than peptides. But do you think that those are worth targeting?
So I can look at it from different angles, but I can at least tell you my personal view. And from my genetics perspective, I know that GBM is a very heterogeneous disease, meaning that you have different tumor cell populations with different type of mutations. And so in the end, I'm convinced that you need a multimodal approach. There's no magic bullet in GBM. I think this has been shown in the past. So the question is how to intelligently combine different treatment approaches. And from my perspective, it makes a lot of sense to combine neoantigens with tumor-associated antigens, like survivin or other targets. And the reason for that is that if you induce an immune response and if you have specific results, you might need some more recognizing additional targets. And you might need to combine even with checkpoint antibodies or bevacizumab, or even some inhibitors that are targeting specific pathways in these tumor cells. So to answer your question from my perspective, I don't have a definitive answer. I think it's maybe not possible. Otherwise, we would cure this disease. But I think in the future, we will have multiomic diagnostic approaches to understand the disease better. And then we have a very intelligent combination of therapies at the right time point.
Yeah, okay, thank you. By the way, why don't we go ahead and start using the raise hand feature now for questions? I see Jason has his hand up, so Jason, go ahead and unmute.
Thanks, Roger. Thanks, Saskia. I'm Jason Binder. I'm a caregiver to my wife who has glioblastoma for about 39 months post-diagnosis and had a recurrence last September. When I was evaluating your treatment, one of the blockers was accessibility and affordability. Are there any plans to make it more accessible and affordable for those in the US?
Yeah, Jason, I think these are obstacles we have to overcome. And what I see is that a fully personalized approach is difficult to implement. And especially when you want to bring this in a clinical trial. It took me really long time to get funding. And when you do a clinical trial, and we are doing this in the US, you need so much money to get to approval that in the end, the therapy will be much more expensive compared to what we have right now. So the first thing is the accessibility. So ideally, patients would get much earlier access to clinical trials. So we need many more clinical trials, and then they need to be financed in a very efficient way so that in the end, the vaccine is affordable. At the moment, it's very challenging. For example, in Germany, we have a very strict process. We have to have a deadly disease with running out of options from the conventional side with evidence of success. We have to go through the process of sequencing, of collecting all the clinical data, of going through an interdisciplinary tumor board, making a treatment recommendation through the team, and then manufacturing a fully individualized vaccine, and then applying it in a patient, which is highly complicated and timely. So I wish we could do this much faster and much cheaper, but at this point, this is as good as we can do it, but it's far from being good.
Do you handle all of those steps at Seagat?
Sorry?
You do all of that at Seagat, both the design of the vaccine, the testing for the antigens, the neoantigens, the administration, the treatment, or do you have to farm that out to other labs?
Roger, thank you for this question, which is really important to me because for me, I have to clarify this. Personalized medicine stands on three pillars. The first pillar is the diagnostic pillar. The second one is the interdisciplinary board. And the third pillar is the therapy. Seagat is a diagnostic company. So Seagat is doing the tumor normal control sequencing from genome, transcriptome. Seagat is doing the neoantigen prediction and is providing these reports. This goes into the interdisciplinary board, and then we have a treatment center, which is actually providing the treatment. According to the German law, I'm the only responsible person, so I have to overlook all processes, and it's independent of any entity. So from the legal perspective, I'm responsible as a medical doctor, and I have to supervise all these steps. So yes, this is all happening here at Tübingen, but only the first part is actually associated with Seagat.
Okay, thank you. Elliot has his hand up. Go ahead, Elliot.
I am a longtime prostate cancer sufferer, which is now in remission, and I also do research on neoantigens. So how would you define a neoantigen, Saskia?
A neoantigen, according to my definition, is a variant that is unique to the tumor and is with high likelihood presented on the surface of the tumor cell.
Okay. When you say unique to the tumor, and the literature is just chock full of those claims, do you mean overexpressed or really unique?
Both. This is not exclusive. If you have a mutation that is important to the tumor, it's frequently also overexpressed. But we distinguish between tumor-specific mutations and tumor-associated antigens. For example, overexpression of genes by definition is not new, like, for example, EGFR or HER2, TROP2. These are targets that are on normal cells, but they are overexpressed in the tumor. If you have a particular mutation that's crucial to a tumor, this mutation can be overexpressed, and you see this in the transcriptome data.
Okay. Given the potential to harm normal cells or antigens that are overexpressed, have you really much experience? Have you ever really found any surface antigens that are really found only either on a particular person's cancer cells or even as a general related to glioblastoma? Something that's really unique, not something that's also expressed in the same way on normal cells.
Well, that's an interesting question because you can have cells that have mutations that are not malignant. And we know that because when I do what I call immune monitoring, measure my T cell response, for example, in my blood, and I don't have cancer, I find T cells that recognize PIK3A mutations, KRAS even, or other hotspot, typically cancer mutations, although I don't have cancer. So I think that every human has cancer-specific neoantigens that come and go. Potentially, the immune system is able to eradicate these malignant cells or potentially malignant cells. And so I think this is not something that's really unique to cancer, but I think these mutations, they should not be there. And if they are there, the immune system should be able to recognize them. And those are the ones I would call neoantigens. And I'm pretty sure that they exist because I can measure T cell responses, specifically recognizing hotspot cancer mutations.
So
This,
how do you get rid of the I assuming that the body is not recognizing it because of the checkpoint effect. Is that the case, or is there some other reason?
well, you know that this is a spectrum. So there could be an initial time where the body is indeed recognizing the mutations, but later on the tumor, when it's growing, is building mechanisms to protect itself. And then you get upregulation of checkpoints, for example. So there could be different stages. And so it's not like black and white, it's a spectrum in my view.
No,
Yeah,
no. Yeah,
you
go
know,
ahead.
on that point, do you use checkpoint inhibitors along with the vaccine typically, or not often?
So again, it's not me personally. So when the tumor board, so we call it molecular and immunological tumor board, which consists of all different kinds of people: oncologists, radiologists, surgeons, pathologists, geneticists, immunologists. If they think that when it comes to determining the right combination for each individual patient, a combination of a vaccine with a checkpoint antibody makes sense, and also all side effects considered in comparison to other options, then yes, this is indeed suggested and done.
Okay, thanks. You know, let's keep moving. Cindy, you've had your hand up for quite a while. Do you have a question?
Yeah, actually, I haven't had it up for quite a while. I hope I'm not skipping the line here, but thank you for calling on me. Saskia, I really think the work that you're doing is important. So I asked this question in trying to sort of piggyback on a couple of things that have been raised. One by Jason, and then, oh my goodness, who's the other person? Well, I guess it doesn't matter. Here's what I'm wondering. Um, the cost of this personalized vaccine, very understandably, has to be high for all the reasons that you pointed out, and it's very unfortunate. And it's very hard for a patient who is, you know, looking for an answer and has really tried everything to think that, hey, there's something here which might, you know, be able to actually help me if I only had, you know, that X amount of dollars. It's a tough situation. What I'm wondering is this, you know, you had said that people come to you with stage 4 cancer, so it's really hard to treat them when they're presenting at that point. So the success rate it doesn't necessarily reflect what perhaps the personalized vaccine might do for someone at stage 2 or stage 3, but at stage 4, it's going to be much harder. And, you know, that makes sense. It goes without saying. I guess I'm trying to understand. Um, You know, what stage of development is this vaccine in? In other words, or this, you know, technology in? Is it, you know, is it reasonable at this point for someone who's stage 4 to think if they could come up with, you know, all the money that's required, understandably, how far advanced is it? I mean, what are you seeing generally? I don't know if you can give percentages, but do we know enough to really operationalize this so someone has at least a 50% chance, 30% chance that at stage 4, or perhaps at stage 3 or stage 2, whatever, that this vaccine will be something that will give them more than just about 6 months or 8 months. So I'm asking you, what is the stage of development? Is this ready for patients? And I ask that because I work with patients. And that's something that they'd want to know, and something that I would want to know before I would say, see, Saskia, she has the Holy Grail. Okay.
Yeah, so I have a vision, and this is driving me. And my vision is that everyone, and I don't have to do this, don't get me wrong, because I'm already working day and night. My vision is that every person, even the healthy ones, are getting personalized, fully personalized anti-cancer vaccines. And the reason for why I believe this is so relevant is because we need to have the immune responses before we get sick. So I actually built a prophylactic cancer vaccine for my husband Dirk and myself. I tested the peptides. I noticed that I already have T-cell responses, and I see for many patients that they do not have a pre-existing T-cell response against their driver mutations. So there must be a time where the immune system is failing, or maybe some patients do not build up these relevant immune responses. So I believe that everyone should have access to a fully personalized anti-cancer vaccine during lifetime. So this is what drives me, but this is impossible to implement. Now, to come to your point, if you have a late stage disease, you have usually gone through heavy chemo and your immune system is down, you get surgery, you get radiation, so it makes it really challenging to build an immune response. And still, in these patients, we see that we can make a difference. We published on a patient with pancreatic cancer, we published on a patient with ovarian cancer, we published on a patient with prostate cancer, we published on the glioblastoma patients, on breast cancer patients. We have a lot of evidence that it is possible to induce a T-cell response in each patient, even heavily pretreated patients. Is this sufficient to help this patient in a particular situation? I don't know. I personally believe the earlier you do this, the better it is. And now you know that it's impossible to have a clinical trial funded, because then, for me, for example, to prove that my prophylactic vaccine is helping me not to get cancer, we might know hopefully in maybe 20 or 30 years. So no one is going to finance that. So this is the spectrum. And now we come back to your patient in an individual situation, not being able to potentially afford treatment. And the peptide vaccine is expensive, but inhibitors are sometimes 3, 4, 5 times more expensive monthly. And so they are approved, but insurance has to cover it, and these are really high costs. So what we have to think about is not only what is most efficient to kill the tumor, what is actually accessible, because some treatments, even when they are off-label, are not accessible to patients, and then they need to be affordable in the combination. In the beginning, I said we need to combine a vaccine potentially with an inhibitor, potentially with a checkpoint antibody, potentially with an anti-angiogenic treatment. This, if you add this up, plus the diagnostics, it's not affordable at all. So I cannot answer your question. I believe that we need to go towards early detection of cancer, towards early treatment with immune therapies. We have to get away from chemo, from therapies that are more destructive. The peptide vaccine has lots of advantages, but also lots of disadvantages. It takes time to manufacture a vaccine. It takes time to induce an immune response. So you can never compare to an antibody that you take out from the freezer and fridge and apply immediately. You have, on the other hand, the CAR T-cell therapies that have an immediate effect that doesn't really last that long. So you have to combine. You have to combine a treatment that works efficiently immediately, and you have to combine with a treatment that potentially gives you a long-term perspective. And there you need to have a treatment with no side effect. And this is a big advantage of a peptide vaccine, for example.
Just one, thank you. Just one follow-up. So in the success case that you've published on, was there anything that became apparent in terms of a pattern of what might have worked in these various types of cancers?
I wish I would know. I don't have a pattern. The only principle that comes over and over again is that it's a numbers game. If you start out with millions of tumor cells, you will always run behind. Then you can maybe control the disease for a certain time, but you will never get control. So if I assume that one T cell can potentially kill 4 or 5 cancer cells, you can calculate how many T cells you need that actually detect or recognize cancer mutations in order to get rid of a tumor that is the size of millions of tumor cells. So the number, I think, is crucial, and the timing of when you come in with the approach.
Thanks, Rick. You have a question.
Yeah, thanks for taking my question. Could you walk us through, sort of a typical protocol for a patient, newly diagnosed and then recurrent, understanding that each patient is unique and that might vary, but for those of us who are a little bit less familiar with the therapy, I think that would be helpful to understand how it sort of works and plays out.
Yeah, sure. So I mentioned the three pillars: the diagnostic pillar, the tumor board, and then the treatment. And so in the diagnostic pillar, we not only do the genome, transcriptome sequencing, we also do a lot of proteome analysis. We do multiplex immune fluorescence. We look at imaging. We look at the clinical data, the lab parameters. We bring in all this data into the molecular tumor board, which is very interdisciplinary, and then the tumor board makes a recommendation. And when, for example, a peptide vaccine is recommended by the board, then the patient comes back to us asking for a cost offer, and then we issue a cost offer, and then patients can discuss this with their insurances, or they say they did, or they have private funding, or did a GoFundMe project, or found money to afford this treatment. We can actually start manufacturing. Manufacturing takes about 3 to 4 months. And then we have a fully personalized vaccine that on average consists of 20 peptides that are all different and formulated into the vaccine. And then once the treatment starts, the patient have to travel to us, to Tübingen in Germany. They come and see the team. They get 4 vaccines in 1 week. We call it an induction, and then they get monthly booster shots. Every 4 to 6 weeks, they get another shot. And after 3 months, we measure the immune response, and actually we repeat the immune monitoring every 3 months. And then we have interdisciplinary meetings where we look at the immune response, and then we make a suggestion on how frequently we would do a booster. We have 14 doses in total. On average, after 10 vaccine shots, we get a plateau. So we come to a point where the immune response can't get much better. And then from there on, we recommend to do immune monitoring on a regular basis, or ideally have booster shots every 6 months, or sometimes even only once per year.
Okay. Chris has a bunch of questions in the chat. Chris, if you want to ask these directly, you can. If not, I'll just read them.
I can.
Okay.
Whatever you prefer.
Why
So
don't
first
you
of all, congratulations to what you have built. I'm very, very impressed with what you have done, and it gives really new hope for cancer patients. And thank you all. Your spouse actually sent to me your publication that you had. That's really very impressive with the outcomes that you had in terms of glioblastoma patients. I have some questions which I hope will also be helpful for the group to understand. So on the one hand side, you do the DNA sequencing and then the RNA sequencing as well. And now you mentioned also that you're doing proteomics. In general, my thinking is that in a way, the transcriptomics is closer to the biology that is effective than the DNA genomics. Do you really then need the DNA genomic testing if you already have RNA or proteomic analysis? That's kind of out of curiosity, and that's my first question. And then in the chat, I have 2 other questions as well, but perhaps we can go one after the other.
First of all, thank you, Chris, for saying that. I think that we need a multiomics approach, and we go very much in the direction of single-cell transcriptome sequencing with spatial resolution. So we really want to understand where the tumor cells are located and how they are, in which microenvironment they are embedded. So we are not only looking at the transcriptome of tumor cells, but also we look at the transcriptome of neighboring cells. And so the transcriptome profile helps us to classify cells. And then we get a spatial resolution, and then we can see the strategy of tumor cells on their way to escape from treatment. So yes, transcriptome gives us a lot of information. I think the bulk transcriptome sequencing is something that we use for the neoantigen prediction, but the single cell transcriptome analysis with the subsequent spatial resolution will give us much more insight in the future. We are already doing this. This is horribly expensive because you sequence the transcriptome of thousands of cells individually, and you need a lot of algorithms and bioinformaticians and experts to get some biological sense out of this data. But I think this is giving us a great promise for the future, and we understand much more how tumors are organized, how they protect themselves against the immune attack and against treatment. Okay. So I think more omics, even metabolomics, microbiome sequencing. So we are extending our omics platforms, and when it comes to transcriptome, we go from bulk to single cell transcriptome with spatial resolution.
How do we get from there to know what the sequence of the neoantigens are? And then how difficult is it to synthesize them kind of on a moment's notice for the individual patients? And I would also assume that there are some neoantigens for certain tumor types that are more common than others. And so my question would be, could we actually shelf them, so to speak, and therefore have a more cost-effective approach for basically to apply vaccinations?
Absolutely, and very good point. This is nothing we do. We are only specialized on the fully individual neoantigens, but other groups have off-the-shelf peptides that they can use while they maybe determine the neoantigens. So you can bridge time by shelf peptides. And like I said in the beginning, I would rather combine strategies than pretending that there's a magic bullet. We have one platform technology, which is the neoantigen platform, but it can be easily combined. It's just a question of money and who is going to do this, the legal framework, and the design of clinical trials in order to show efficacy of a multimodal approach.
Yeah. And then I probably I then have a final question on time and costs, and it's actually I had yesterday a conversation with a melanoma patient whose Sage Oncotest actually showed that the tumor is basically multi-drug resistance against chemotherapies, which is not surprising for melanoma, but also against BRAF and MEK inhibitors as well. And so there is nothing in the targeted and chemotherapy and targeted therapy that actually could work. We were very pleased to see that there are some off-label drugs, in this case disulfiram, that actually had anti-proliferative effects, very strong anti-proliferative effects. I was actually excited to see that we at least found something that gives this patient hope. But I actually, I think that the next step will actually be the normal PD-L1 inhibition for this patient. But I'm actually wondering if this patient would be interested in your neoantigen vaccine approach, how would this work? And the question is also, what would be the approximate cost for this patient?
So the first step when a patient is approaching us is to generate the data, including the sequencing data for genome and transcriptome, and to look at the neoantigen list. So this is what Seagat does. This takes about 4 to 6 weeks, and depending on what we include in terms of tumor boards and looking at imaging, and this is in the range of 10 to 20,000 euro in order to get to the point that we can access a tumor board that would be in a position to bring a personalized vaccine into a clinical context for this particular patient. And then when the tumor board recommends that a neoantigen vaccine from our platform would make sense for this patient, then the cost is maximum 60,000, additional 60,000 in order to manufacture 20 personalized peptides, to have 14 applications in our center here in Tübingen, and that also includes every subsequent tumor board, immune monitoring, multiplex immunofluorescence, and other diagnostic tests that need to be done during the navigation of the patient through the vaccine journey.
Is that $60,000 or euros or?
Bitcoins.
In this range.
Okay, okay, close enough. Okay, thanks very much. Karen, you've had your hand up for a while.
Hi. Wow, you have said so many interesting things. I'm just now remembering my question. So I was intrigued that you said it was a numbers game. Could you comment? I would naively expect the relevant T cell to expand quickly. So I'm a little curious about that, and also just out of curiosity, and also given that it is a numbers game, could you expand the T cells ex vivo?
Excellent questions, Karen. So I wish we could calculate this a bit better, and I think it has a lot to do with accessibility of T cells to cancer cells and the microenvironment, because we know also when T cells expand and they get a very unfriendly environment that contains a lot of lactate, for example, and a type of macrophages that we call M2, they very fast exhaust. So this is not only number, but it's also about activity and exhaustion, and to see whether these T cells are in the end efficient. And yes, there are approaches to expand T cells. So you can, if you have the platform, you can, once a patient has developed a peripheral T cell response, you can do a leukapheresis. You can get the T cells, you can expand them by stimulating them ex vivo with an individual peptide or even a peptide pool, and then you can activate them ex vivo and then reinfuse them. So technically this is possible. I actually have been with a patient who has undergone such a procedure. It's nothing that I would say is doable in an easy way, but technically it's possible. And it's an interesting approach.
Yeah, thank you.
A question from Mass Medical Angels about the adjuvants. I assume you're using an adjuvant with the vaccine. Is that right?
Of course, yeah. Peptides per se are not very immunogenic. So we are using GM-CSF. It's called Leucine in the US. It's FDA approved, and we use imiquimod topically. So Leucine subcutaneously and imiquimod topically. And this is also described in our publications.
Do you get strong side effects from that, or not so much?
Not at all. I mean, we sometimes have allergic reactions, and they can occur over time, and we have to closely monitor them. The skin is actually showing us very nicely how we can go from no reaction towards more redness and itchiness, more reaction towards the vaccine over time. Sometimes when we do a vaccine on the right side, we see the bumps coming up on the left side. So the skin is actually a very good organ to observe the work of the immune system.
Yeah, I asked because I did a neoantigen peptide vaccine, and I would get flu symptoms. And most days they were super mild. One day it was just really, it really knocked me out. So Darren has another question. Go ahead, Darren.
Yeah, might be several questions actually. Maybe take it one at a time. We've been working on access issues in America, and Vanessa Hugo and Jason Binder also in here. There's a filing you can file the FDA called a single patient IND, and I've met a variety of neurosurgeons that are willing to fill out this paperwork. It's essentially a single patient clinical trial for one. And the Jamie Lander Foundation and others have worked on that paperwork. Willie Hoos in particular on getting that paperwork to be easier to fill out. So one question I would have is, why not have some American file a single patient IND with their neuro-oncologist and with the FDA, and then essentially mail it back and forth, just because the you could imagine in a world where there's only so many advanced processing centers to do the target selection and the print out the peptides and stuff like that. Maybe we should be doing this by mailing back and forth.
Well, it's a legal problem that's not happening in the US, but in Germany. In Germany, we have a specific framework which is called an individual healing attempt that allows a medical doctor to do a treatment that's not approved. But you are not allowed to send it out. So the IND filing, the individual filing in the US isn't helping me. So then you need to find a partner in the US, like the Jamie Leandro Foundation, but I cannot be a partner. What I'm trying to do is to run a clinical trial. And so we are very close to starting phase 1 for GBM. Very soon we will have our publication out on IDH mutated brain cancer. I want to start a clinical trial for those tumors. I'm seeing children with H3 mutated diffuse gliomas. We have the BRAF mutated brain cancers. We would like to do a phase 1 for pancreas, for colorectal, and so on. So we can actually run through all the tumor entities once we have sufficient funding. And this we would do in the US. But we cannot work with individual patients yet, like you describe it in the US.
I could, do other questions, or we could just keep my hand up and wait, have some other people have some questions.
Yeah, Jason, why don't you go ahead?
Dr. Biskup, what can I or the brain cancer community in the US be doing to help you besides finding funds? Like, I'm going head to the hill next week. I'm connected to a lot of folks in the US who are trying to destroy GBM like you. If you have a promising thing, like what could I have in the back of my mind that could be helping y'all's cause?
Thank you for asking this, Jason. So our plan is to start with private placements now, and then we will try to find investors for Series A funding to really lay out the phase 2 for glioblastoma patients, GBM, and then we want to go public. And at the moment, it's, as you know, a difficult time in the US, but we hope that once we can maybe go public, we can have many smaller contributions. At the moment, we need a few people believing in the technology, helping us to do these initial steps. But once we get going, I think we will have potentially many more people helping us with smaller amounts of money in order to get to the approval. Once we have approval, I can assure you I'm not taking the money for myself. I will put it in the next clinical trial. And so this is the way that I'm trying to do this now. But the more you connect, and we have many friends and believers at the moment. And I think it feels a little bit like a tipping point. I've been working on this for so many years, frustrating evenings, weekends, so on. And now it feels like we come to a point where many things come together, and maybe as a community and maybe as many partners, foundations, we speak to so many people at the moment. I hope we come to a point where we get this going faster. But thank you so much for asking this question.
A question from Mass Medical Angels.
A hand raised question or in the chat?
Yes, go ahead. You're on.
The hand raised question is I don't want to spend too much time on this. I'm not a patient, but when you mentioned a private placement, I just wondered how much are you looking for? I know you're not soliciting funds in this meeting.
Sorry, let me stop you there. I don't want you to do an unlawful solicitation of securities. This is a
understood.
Forum, so we can't talk about that, but you can do that.
Okay,
offline.
Understood. I tried to be careful. Is there time? Could I send you an email?
Yes, please.
Okay. Thank you, Roger. You're probably absolutely right about that.
Brad and Chris, I think a few people have my email address.
We'll circulate that. Elliot, I know you had a question earlier that we didn't get to. Do you want to? And then we'll close with Darren.
Yes. So most of these vaccine type things focus on T cells. Has there been any investigation on the B cell response?
Wonderful question. I wish I would know better. I think there will be some antibodies that we potentially will be able to detect in the future, but we do not have the technology at this point. But if I would have to bet, I think B cells will have a role. When we do the multiplex immune fluorescence on those slides from tumor tissue, we very rarely see B cells in the immediate area of the tumor, but I think it can be a systemic response, and I think especially the communication between immune cells is crucial. We know this from infectious diseases, and I think it will be very similar, yet I don't have the data to answer this question at this point.
You know, a question just popped up in the chat, and I was going to ask you about this as well. You were talking about the tumor mutational burden. The question here is, does the vaccine respond to cold tumors the same way as other immuno-oncology agents?
We need to better understand what it means immune cold. You know, there are differences in how the tumor microenvironment is structured. And I mentioned the spatial transcriptome analysis, and I think we need much more data, and this is also, again, individual. So for me, the puzzle becomes more clear when I have the spatial resolution of tumors, and then based on this information, I think we can make better suggestions, and potentially we might need support by machine learning and other algorithms in order to understand better what will be the killer of this particular disease.
Okay. Darren, you get the last question.
Thank you. We have a number of groups in America and Mexico doing somewhat similar things with a certain software stack. I just mentioned the Pan American Cancer Institute, Matt Halpert at Immunoscene in Cancun, and MD Anderson, Roy DeSouza and David H. Hawk at Break Bio. But yeah, Delia Hormigo, especially we had a meeting with Delia Hormigo at Albert Einstein Medical College doing GBM and the sort of peptide vaccine. Is it possible that we might, take the patient and not, necessarily do the medical care through the mail, but take the sequencing information and have everyone's software stack pick the targets, sort of compare on target selection to say that, these different groups at Mayo Clinic and whatever, ran the sequencing through and decided these were the top targets that are most likely to really cause a strong response, and then do that kind of comparative analysis to say that, for this one patient, the different groups found different, target predictions in a different sort order of what was most likely to be effective. Is it possible there might be a cooperation of that, like on a data level rather than on a treatment level between the different groups and countries?
So first of all, we are sharing data. So each patient has the right to access his own data and can share with everyone. So this is done daily and important, but it will not move the field in large steps. It's only incremental, I think. The larger steps will, I'm convinced that this is the multiomics approach. So we frequently see that, and I can say this as a geneticist, that the genes it's not everything. So you need transcriptome, you need the proteome, you need the spatial information, and you need metabolomic information, metabolites, and you need the microbiome. So it's much more complex, and sharing DNA and transcriptome data is not moving us really fast where we need to go. So that's what I think. And I think as a geneticist, I can say that.
Okay, thanks very much. So I know people may want to follow up, may have additional questions. We're a little over time now. So I will drop, unless you have it, I'd like to drop your email address into the chat if that's okay. Or how should people contact you, Dr. Biskup?
I can put my email address in the chat. I'm most happy to receive emails and further questions, and I think this is what's really moving the field, exchanging our knowledge, combining human intelligence. So thank you all very much for joining. It was a pleasure to speak with you, and I hope we will be in contact.
Okay, thanks very much.